Welcome to Face Space Invaders. I'm happy to say we got to the 20th interview on the podcast. Today, I'm talking to Rommie Amaro, professor of chemistry and biochemistry at the University of California, San Diego, who works at the intersection of computational biophysics, drug design, and microbiology. Her research was particularly impactful in the context of the COVID-19 pandemic, when the Amaro lab made use of their extensive experience with viral glycoproteins to model and investigate the structural role of glycans in the spike protein, responsible for cell recognition and evasion. After that, they kept delivering fascinating results within the field of computational virology, including a model of the entire SARS-CoV-2 virus. So, out of curiosity, I initially asked Rommie how this enormous project first unfolded during the early days of the pandemic. And from that story, we moved on to discuss her original motivation to study large complex systems. We touch upon the exciting experimental developments that enable the realistic modeling of systems as big as entire viruses. And we highlight some unexpected findings that came out of the large-scale simulations. Rommie also shares her reflections about the collaboration-driven nature of her lab and the complexities involved in interdisciplinary communication. And we wrap up with a few thoughts about the AI-driven brain drain from academia to industry. We're again digressing a lot, and I'm sure you're going to enjoy the breadth of Rommie's insights. Let's go! Okay, so Rommie Amaro, welcome to the podcast.
Hi, thanks. Thanks for having me.
So Rommie, in recent years, you've been known for your impressive work on large molecular systems, and a large part of that relates to your efforts in understanding the structure of the SARS-CoV-2 virus that caused the COVID-19 pandemic. And that, as I understand, was a consequence of your previous research on the influenza virus and tropical diseases. And can you share the story of how you and your lab turned this, unprecedented global crisis into a nevertheless impressive learning and dissemination opportunity to the point where the figures from your simulations would make it to the newspaper covers?
Sure. Yeah, that's a good entree to the discussion, I suppose. So, reflecting back. Right. So my group had been working with viral glycoproteins for some time. I actually sort of fell in love with influenza neuraminidase when I was a postdoc with Andy McCammon. I just thought that it was such an interesting protein with its dynamics and, all that it did. So continued to study that, and also hemagglutinin, and then, have always been interested in sort of pushing the boundaries of simulation and sort of like trying to get towards complexity. So naturally evolved to working towards simulating, the whole virion for influenza. Right. And then we were sort of plugging along and getting close to publishing our first big paper on that. And that was right around the time when the pandemic hit. And it sort of I think it all came about fairly organically. I mean, as soon as we started, I remember in December hearing about this, virus in Wuhan, and people already sort of, becoming alert to it, sort of watching it. And then in January and so forth, it getting worse. And then in beginning of February was when we started thinking maybe we probably should start pivoting towards this. And then the structure came out from the group of McClellan, and we started working with it immediately. And in fact, as soon as the structure was available, I emailed the director of the Texas Advanced Compute Center, Dan Stanzione, who, has been a longtime colleague, sort of anticipating that we were going to need some big compute because it was going to be a beastly system. And we know most of these are. And Spike in particular turns out to be quite large, the largest of the glycoproteins that we've studied. And we've studied a whole bunch now. So, and he was just super supportive from the get-go, from the very first correspondence, was absolutely, we'll, do whatever we can to help you. And I think that whole attitude was something that carried through for the first, number of years of the pandemic, at least for the first 2 or 3. It's kind of changing now, but it was just a remarkable opportunity. And I mean, one of the things that I always say, or that I've said before, is academia in general and science, sort of the pursuit of science, has a lot of weird things about it, in terms of the ego and the competition that we sort of set up, that we construct. And just the whole way the whole system is constructed is toxic in a lot of ways, and sort of like what is incentivized and what is disincentivized, even if not directly, indirectly, and so forth. But what the pandemic really did for folks was, it sort of put us all on, in some sense, even footing. I think in some way, it like opened people up to really solving a problem together, as opposed to, I think for the most part, and of course, I'm sure there were people who, really were still thinking about it, maybe possibly as a way to just like leg up or something. But I think people really genuinely, for the most part, all the ones I interacted with, I should say, were really genuinely interested in trying to make progress for humanity at that point. And that was really special because it disarmed people in a way that I think I had never experienced before. And all these things, all these incentives and all these reasons why people otherwise sometimes find themselves doing science, these ways that people work towards solving a problem, were then just dismantled. And then at its core, what one is left with is just human beings, trying to do really kind of cool science and to work together. And then not only did you have that attitude, but then it was also the mobilization of any resource that you needed, because it was really the whole world wanting to move forward. So, we had just a really sort of special, remarkable moment in time to like press on a scientific problem in ways that I'd never really done before. And so, I don't know. It was crazy at the beginning. It was exciting, but it was crazy. It was really exhausting. I think that I know for sure that people in my group have PTSD, and me too.
Many groups have gone through this. Yeah.
Many groups. Yeah, it's probably true, right? Like, I think we kind of all do, and are sort of still in some ways kind of like dealing with it or grappling with it and the change and sort of like moving on. But like, in my group, it's also kind of weird because like, we were studying viruses before and we're just like genuinely interested in viral glycoproteins, right? Mm-hmm. So we're still interested in like, how do these things work? There's so many questions about membrane fusion and intermediate states, and there's all this good science. But in some ways too, people, you hear people say like, oh, SARS-2, it's over. Like people are really kind of getting tired of it. But, still in terms of the science, it's like, oh my gosh, there's still so many cool things that we don't know. So now we're kind of in that phase, which is also a little weird. Like, are we moving on? Are we going in deeper? Like, what are we doing? But I guess that sort of strays from your original question. But I think it was just this amazing, this amazing kind of confluence of different factors.
Yeah, I think it was an amazing moment, right? Because people could see both the kind of nascent science of it, right? Because there was no or very little science of that virus, mm-hmm. aside from maybe the previous virus that was there before. And also that, for virologists, it's such an unprecedented opportunity to trace something on so many levels, right? It's like the genetics of it, the structural evolution. So if you happen to be from that field, like computational virology, which I remember it was a very kind of tiny and cryptic field back in 2016, 17. I mean, a couple of years prior to the pandemic, it's like maybe it had a session at the BPS at the conference, right? But then there were a few people doing that. And suddenly it thrust the field into the mainstream, right?
Yeah, no, definitely. I mean, it was neat too, though. Like, you had people who maybe they thought about viral glycoproteins or maybe they didn't, but that there's so many different types of people, even in computational biophysics or computational chemistry or computational biology, however you want to define us, there are people with so many different skill sets, right? Like the force field developers, the algorithm developers, like the people who know whatever it is, like But it was a time where, again, people were coming with all these different skill sets, like towards one problem and being like just super open to collaborating. And, and like the model checking. I mean, when we built even just that part of it, like when we built the initial system, I mean, it was really a nightmare because it has like all the protein and some of the protein was missing. And so there was a whole issue with like protonation states and which, cysteines are going to be bridged and which aren't. And maybe it depended on the conformation of a loop and this and that. And then you had, all the glycans and everything. And it was a lot to make sure that it was done correctly. And it really, I think we benefited tremendously from the input and the care of colleagues, like, for example, Carlos Simmerling. I mean, he was like looking at our structure like at 3 a.m. and saying like, actually, I think you missed this one position over here, but it was like that from like so many different angles and from so many different people. And that also just like helped to advance the science a lot. So yeah, it was a crazy time.
Right. This integrative angle was impressive, right? Because there's always this question. I think it was a big question back before the pandemic. What are we actually learning from large systems, right? So, okay, we can build systems, we can run simulations on them, but what is the information we're getting from the simulation, right? That we didn't explicitly put in. And so that's my question to you. How do you see that these days? So what are the new things we have learned from running those huge systems?
Yeah, I think that, first of all, that's sort of like an interesting question. It has, in many instances, I have to say it's a question that can be almost weaponized against folks who have had an interest in big systems, and that goes back to Klaus and others, where it's like, oh, but what are you really learning? And I think actually there's been a lot that has been learned, and I can go through, the details, but I think it's also, and just because I have to do this, because, I went through Klaus's group and I worked with him, and he's not here to say this, but so there's also something to being able to do something just because we have an ability to do it, and not necessarily knowing in advance, this is the exact hypothesis I want to test, but understanding that when you can bring all these different components together, put them in the mix, that you're going to learn new things. You're going to have new vantage points. I mean, it's very similar to sort of climbing a mountain. I mean, why do it? I mean, it does in some sense, it brings you to a new level and gives you an entirely different perspective on problems, that you have maybe been looking at from, one perspective. And so I think it is just important to have folks around who, and it does take all types. So I'm not saying like everybody should do big systems, but I do think the pursuit and study of ever larger and more complex biological systems is something that is important and sort of should always have a place, and hopefully, is appreciated by folks. But I can say now too, with much more confidence, as we know more about structural biology and about proteins and molecular machines, is that the dynamics that these systems undergo in situ, in cells, is different than what they can study in the bench, in vitro. It's the stuff, all the single particle cryo-EM. It's amazing. But is that landscape, are those conformational dynamics the same as what's actually happening when it's in the ER or whatever? I mean, actually, pretty sure it's not. It gives you a lot of it's a wonderful model system. It's really important for understanding of structure. There's all these amazing advances, right? But putting it into context is critical for actually understanding true biology.
Right. It's interesting that you bring it up, because I think there's this kind of, I hesitate to call it a paradigm in science, right? But there's this other mode of approaching science, which is by observation of something that's a complex system. And this complexity science is becoming a proper branch of science these days, I think. So I think what you're advocating for is exactly this, right? So put together something that's complex beyond our simplistic imagination, and then observe it with a different toolbox without a prior hypothesis or prior expectation of what it's going to do.
Yeah, I mean, I think that's certainly a large part of it. And, the funny thing is just, again, how the whole scientific system is constructed. I mean, like review panels, we tend to be more narrow. We tend to not want to do the edgy kind of high risk stuff. What are they going to learn? Is it really going to be worth it? I mean, but now, looking back, I don't know. It seems to me like, yeah, it definitely is worth it. All the work that we did to construct, for example, influenza, and before that Klaus had been doing, at the all atom level, and there were certainly others. I shouldn't just focus on him and my efforts. Understanding, of course, that we're part of a very large community. None of this would be possible without all the different pieces, of the puzzle that make it work. Who we're deeply appreciative for. Just that all that work that had been done with HIV and influenza, it was because of all that took a decade to develop the tools where people really questioned, and like the stuff with influenza, we had real trouble even getting the compute time for that. And it wasn't until Klaus basically said, he kind of took me under his wing and he said, hey, Rommie, I believe in what you're doing. I've been fighting these fights for years. And I'm going to give you some of my discretionary time on this big fancy machine we have here at NCSA. It's called Blue Waters. And, you can't have all my time because I needed to study this and that with HIV, but we'll work with you. We'll support you. And that was just super critical to getting the preliminary compute that, allowed us to develop the tools and stuff or whatever. And by the way, we did learn amazing things about influenza, which I'm happy to chat about. And it does have to do with dynamics of these glycoproteins that you don't sample necessarily or in the same balance of, states in the crowded environment.
Sure, we can dive into that.
Yeah, versus the sort of dilute solution type stuff. But it was all of that allowed us to, be ready to go with SARS-2. And so, a similar system that had taken us 8 years to actually build and get to production dynamics, we literally did it in, it was running by November. So it was 9 months? We did it in 8 or 9 months. I mean, I don't know. Yeah,
that sounds like part of serendipity, right? That you can be lucky, but only if you're well prepared from prior work you've done.
Right. Mm-hmm.
Right. So let's talk about what you learned about influenza, because that's a great case for those big systems, right, to be made.
Yeah. I mean, what we learned, and which I think we mostly already know, but the issue is that when it comes to structure, I think the issue is that when we come to structure, we are just only now developing the capabilities to actually see what happens to these systems, again, in situ with all of the complex interactions of the environment. And even then, it's still static. Okay. And I'm thinking of cryo-electron tomography, for example, with the milling, or where you can do subtomogram averaging on these systems that are really sort of in cells. But anyway, so what we learned with the flu, and my group had studied, and others for years, a decade, had studied these hemagglutinin systems. So hemagglutinin is the class 1 fusion protein for influenza. It's a trimer. It has a lot of similarities to the SARS-2 spike protein, to HIV envelope protein, you name it. And, it's interesting because it's a target of vaccines and so forth, on top of being mechanistically interesting. But anyway, so we had studied these systems for years and looked at, binding of various groups and so forth. But when we put it in the context of the crowded virion, and, all of these viruses, they have different propensity, different numbers and density of the glycoproteins. So SARS-2 has maybe about 40, HIV has 10 of these glycoproteins. Influenza is the opposite. It's super packed. It looks super, super crowded. It's got 200 and some hemagglutinins, and then another. Yeah, it's got a bunch. These guys can basically hardly move, it looks on the surface. So we built it, got it all in there, simulated it. And what we saw was just stuff. We were seeing motions that we had never seen before in these, in all of the many, probably now hundreds of microseconds of dynamics, of single particle dynamics. We never saw these types of motions. And these types of motions, what are they? They're a really kind of more extreme tilting that happens relative to the membrane normal. And then there's also breathing that happens. And the breathing, it turns out, has to do, so I think these molecules, as we know, they do actually breathe on their own. But when they have, if they're just like, again, in dilute solutions, of course, you're going to have these different intermediate states. But the structurally resolved state, the one that they could get to the high resolution, was always the tidy, upright, fully packed trimer. And so I think that colored how that data, that was just like such a beautiful picture. It totally biases scientists about everything, about how they think about it, about how they interpret how it's interacting with things. But it turns out that it's just much more dynamic than that. And when you put it in the crowded environment, we actually see sort of very quickly evidence of this additional dynamics, including tilting, but also a breathing open of the protein itself. And in the case of hemagglutinin, influenza hemagglutinin, what was interesting about that is that when it breathes open, there's a quote unquote cryptic epitope, cryptic being a fun word for things discovered through simulation that weren't there in experiment, a hidden epitope inside of hemagglutinin that actually could be a viable strategy for neutralization. But that sort of really was a curious finding because they weren't sure, like, well, how does it really get access to that and so forth? And it turns out it's part of it, sort of like ordinary breathing, which we saw and which was actually substantiated by experiment also, but at low resolution where you can just see sort of like the blobs opening.
Do you also have knowledge of the higher level? Yes. I recently had a conversation about, like, do we know what happens to a virus when it dries up? Let's say you have a droplet, right? And it evaporates.
Well, like
why does
that's
a
another
virus,
whole interesting story.
Yeah. Why does a virus get deactivated?
Yeah. So, that's a question that we're trying to understand and why, and we don't have we have hypotheses only because we have these big simulations, but we're still trying to understand that. I think we're getting closer, but I don't want to spill any additional beans. But it looks like, I think things that I've said before, it does look like for SARS-2 that in these like respiratory aerosols, right, which are pretty much the predominant mode of transmission for this virus. And that whole sidebar of interest is like, why are we so reluctant to admit that it is airborne? And it's just mind-boggling to me. But in any case, understanding the physical chemistry questions of what's happening to these biological things in aerosols, that's like a whole frontier field that is just beginning to get cracked open. And one of the also kind of weirdly serendipitous sort of things was in 2018 at UCSD, where I'm a faculty member, there was this really big NSF center. It's called CASE, Center for Aerosol Impacts on Chemistry of the Environment, which was studying sea spray aerosols. So, like when we're really close to the ocean, gratefully, it's beautiful here. And the ocean, the waves crash, and then like small bits of the ocean actually become aerosolized. And these bits of ocean, they're not just like salty water, right? Because the ocean is like filled with all sorts of critters and so many biological things. So there's all sorts of things that actually do like get transferred from the bulk ocean into the atmosphere. And they can do things like seed clouds, react with atmospheric gases. But so we had studied separately ways to use the quote unquote computational microscope. In other words, these really larger scale MD simulations to study what happens inside these aerosols with this sort of like complex mixture of different kinds of fatty acids and ions and lipids and all manner of things. So when COVID hit, we were not only studying, the bulk of my group is studying like biomedical research type questions, viruses. We also work in cancer. I have a whole drug discovery branch in the group. But there was a small bit, a few researchers who were working on aerosols, these sea spray aerosols, which were kind of viewed as being disconnected from health. And anyway, that gave us the tools then to converge respiratory aerosol chemistry with viral chemistry, which turned out to be, it's turning out, of course, it's like really critical for SARS-2, but also many other, pathogens. So, yeah, that gave us a platform to develop our like uber crazy, insane big simulations that were like over 1 billion atoms and were just crazy, you know, or we had all the complexity of the respiratory aerosol that has like mucins and different types of things coming from lung surfactant and all that.
That's an impressive coincidence. I was just thinking, if you use coarse-grained models, how you simulate evaporation, but you're saying you're actually doing this at a fully atomistic level.
Yeah. So we did it. Evaporation is, of course, interesting. So we generally tend to assume a type of a relative humidity, which gives us
Stability.
sort of a,
okay.
yes, a local stability of the aerosol, you know, and there have been folks who have developed fully atomic sort of ways to look at evaporation. And I should also say that in these, so we have the aerosol, which is like all packed, but then it's also, we're not necessarily using periodic boundary conditions per se. Like there is an air-water interface in this box, and you do see escape of water molecules, but it's not, but it's a minor fraction. And so it's, you know, it's not actually evaporating in the sense that,
I see. So you're
dramatically changing.
Mostly looking at the partitioning between the bulk and the interface.
Yeah, we're looking at morphology, what's going to the surface. And then in particular, also, you know, so for questions of sea spray aerosol, we're interested in the reactions that are happening on the surface of these aerosol particles or micro droplets, which is interesting because the chemistry totally changes at the surface versus in the bulk. Reactions get dramatically accelerated in some cases, or like, you know, for various real sort of detailed chemistry reasons. You know, in the case of aerobiology, which is maybe how I would call what we're doing now with SARS-2 in these droplets and aerosols, it's more, you know, the questions are more about what is happening to the structure of this thing, because these are fairly small particles, right? They're basically on the same order of magnitude size of the virus itself. And we know from structural biology that at the air-water interface, structure will be degraded. It will be warped or whatever, right? I can't find the word right now, but it'll get messed up. So there must be something protecting the virus in flight, or at least some proportion of the viruses enough that, you know, they actually can remain infectious. And so these are the kinds of questions that we're curious about what's happening in aerosols.
Have people studied those droplets like aerosol particles with, is this even studyable with, let's say, in situ cryo-EM?
I know that's something that, well, so there has been one study actually also coming out of UCSD where they took a sea spray aerosol and they trapped it and were able to visualize it. This was sort of, I think, one of the larger, it was probably more the size of a droplet, not an aerosol. Okay. So aerosols are like the little ones that are, you know, they have to be about a micron or so in size and smaller. And then beyond that are droplets, and those guys are bigger and then they fall off, but it's the aerosols that keep floating. Anyway, so there are folks who we're trying now to develop techniques to trap and visualize these actually using tomography, but that's still something that needs sort of methods development.
Yeah, I was going to ask, is there any kind of secret experimental sauce that you can use these days beyond cryo-EM tomography that helps you study those things? Like what are the latest improvements in experimental techniques that make it doable?
Yeah, there's you mean for aerosol chemistry in general?
Well, it's virology
basically
broadly.
Yeah, I think one of the most exciting places right now to do this type of research is actually at the University of Bristol. There's this professor there, Jonathan Reed, who has been working in aerosols for a long time. And they have ways to basically, using something called like an electrodynamic balance, they can basically like trap individual aerosols and then like interrogate them under different conditions. Oh, wow. Yeah. And what's really, to me, the technique is really cool. You know, it's an amazing experimental technique, very unique.
So they're balancing the gravity with electrostatic fields. Right?
Yeah, fields.
Okay.
Yeah.
Cool.
And so it's really neat. Yeah. And you can make this sort of like almost direct connection. They can't see structure per se, but they can look at so many other things. And to me, that's also exciting because I think we don't always have to see the structure necessarily, right? This is where computing actually and computational biophysics actually should be really complementary and useful to sort of poke at and go into places where experiment can't go. That's some of my favorite things to do. And, maybe the experiment will catch up later. Maybe we'll be right. Maybe we'll be wrong. But like, can our hypotheses stand up to different types of experiments and, advance our understanding in the now? And I think we're able to do that. But anyway, yeah, those methods in particular being developed by the Reed Group and so forth are really cool. And the other thing I want to say is that they've done a really good job at intersecting the disciplines required to make those successful experiments. Because I'll tell you what, I think the hardest thing about science is not, and here's getting a little bit on a soapbox of like, what do you want to talk about that's not necessarily science? But like, I'll say this, I don't think it's like the technology development that's like the hardest part. I think it's like the people that is the hardest part of science. And that can come in different ways. But I think like when we were starting to work with aerosols, we stepped into, we were always working in computational biophysics and looking at enzymes and proteins in cells, blah, blah, blah. And then we go over to this other part of the building and we're starting to work with really large groups of really smart people who have a completely different vocabulary and are coming from a completely different like view of systems. So like I say substrate, and I'm thinking of one thing and they say substrate and they're thinking of something completely different. I say residue. There's just like, it took us 2 years to even be able really to have the kind of conversations where you can go back and forth and you can really understand each other and you can trust each other enough to do the experiment. And, and that whole aspect, that's actually really hard. And it's also hard to just have that in a science session. It's even harder to have that in a review panel. So what happens is science becomes siloed into these like niche groups where everybody can understand each other. But then you do have people and groups who are working at these interfaces. I personally think, and many others will agree with me, that these interfaces of science is where all the interesting problems are. It's where the new solutions are going to happen and where we can actually address sort of frontier things. But it's like, it requires really hard work just from the people side.
No, I absolutely agree.
I think that agree. Good.
Is there a case to be made for, like a transformer style translator between subfields of science, right? Like you would see what the other person is saying in your language, like computational biophysics language in real time.
Right. Like, had some kind of software that would real time translation of like, what are they actually saying? Like in my, maybe like decade from now or so.
No, I've experienced it myself. Yeah, so many times that after many, many meetings, you spend another 2 hours like going through your notes and trying to see what these words mean in that community.
Yeah.
Okay. Talking to AI people or talking to visualization people or talking to, proper experimental people. Yeah, absolutely.
Yeah. And it's
someone
funny
should work
like,
on this.
They should. There's so much good things to be done.
But on the people side, I actually asked people, what should I ask Rommie when I talk to her? And a few of them,
really?
I'm always curious about people's input. And a few of them pointed out, and I was similarly curious about it. How does it happen that so many of your group members gain so much visibility, right? I'm talking, let's say, social media, because people like Fiona, Kieransa, like Lorenzo, like Mia Rosenfeld, like Mohammed, I've known the people from Twitter, for example, right? And then I see, they're actually connected to your group. Do they just get inspired from you, or do you select people who have this kind of personality that they're outgoing?
I see.
Because it's interesting
that's a good question.
It happens a lot, apparently.
Well, I think I don't think everybody is necessarily the same. Fiona had like a big following and she's super active on social media stuff. I think like from well before. I only joined social media type stuff in 2020, randomly also right before the pandemic. Right. And and then there's like Mo, and he has like a whole YouTube thing. I don't know. Are they drawn to my group? I don't know. Lorenzo joined before. He might have been on a social media thing. I don't know. So this is an interesting question because I've never thought about.
It's not a lab policy that everyone gets social media training.
No, God, no. And I think No, I'm hanging on to social media by a thread because like this whole transition from like Twitter to X has been a nightmare, and I've become kind of like just less active on the platform. But I think that for sure in my group, we do a bit of methods development, but we do have the bulk of our projects require extensive teamwork, and not just like a collaboration with a group, but like if you're working on a project in my group, it's very likely that you're going to have 5, 6 different collaborators with different experimental techniques converging on the problem that you're studying. And so you have to be able to work in teams. And that means in some sense, usually a very good communicator, kind of probably a little bit more on the assertive side. And maybe that's why it sort of also trends that way. But at some point too, I'm sure it attracts a certain like type or kind of person. So maybe that's like even
chicken and egg. Yes, I see. But yeah, that's a great point, because it's not always that common, right? Putting people in this collaborative environments. Not all groups do that all the time. So
definitely not. And definitely not academic groups, and definitely not chemist academic groups. Yes, it can happen, but I feel like my group is a little bit different than some of the other groups that you would find in a traditional, say, chemistry department, where the questions are maybe a bit more tightly focused. We sort of bleed across from small molecules to proteins to larger systems now. Mm-hmm. And so there's interplay, strong interplay with other groups.
More room for exploration as well for individual.
Mm-hmm. But I have to say, I also definitely have people in the group who you've never heard of or, who have like zero presence.
I can imagine.
Yeah. And
no, I know you have a big group, so I don't know everyone for sure.
Yeah. And it's not like, it's not like everybody is the same for sure. I mean, we have one of the things that I've always taken a lot of pride in is the diversity of my group. I sort of was raised thinking that diversity is a huge strength. And it doesn't matter if you're a sports team or if you're a family or if you're a lab. Having different perspectives is like one of the most like important and I think beautiful and interesting parts of life. I think the Amaro Lab is a place where you find that type of diversity, whether it's geographic, whether it's religious, whether it's even just like kind of almost also like what interests you. I have people who are like really interested in something very deep and narrow, and then others who are like, oh my God, how does this amazing thing work in this big sea of complexity? So I don't know.
Yeah, many voices.
It's a diverse group.
That coincides with what Saima said before on the podcast. So I'm happy to spread this message.
Yeah, she's someone, it's funny, there's also all types of colleagues, right? Saima is someone who I've enjoyed meeting and who I think we share. He's a bit of a kindred spirit. You sort of like, you run into folks and you feel like sometimes like either I've known you a long time or somehow, like you just really kind of get on with them very well. She's certainly one of those folks. Yeah, she's a keeper. She's a gem.
Right. That happens once in a while. That's beautiful.
Yeah.
So do you have any final thoughts you wanted to share with us about the drug discovery side of your research?
Yeah, I have to say I want to stay kind of positive because, I've worked for many years at the intersection of simulation and sort of drug discovery and design. And, had sort of my first part of my career was really all about methods and applications, connecting structures from simulations with small molecule screening and so forth. Started a company, have a lot of patents. I think it's an interesting moment, for this field, sort of looking at the small molecule side with like this AI wave. I think one of the things that I find unsettling, I think I can use that word, or that sort of sometimes maybe keeps me up at night. I don't know, maybe it shouldn't bother me, but like a number of my colleagues have sort of gone the route of leaving academia, and trying to run in the herd with all the AI venture startup and all this. And I think that that's, normal and good. I also worry a little bit about the academic side and about methods development and about, really investing in problems where the solution is not necessarily right around the corner, but that requires, a real kind of slow chipping away. I mean, there are definitely problems, like that. And with the sort of the money being thrown around, it's attractive to jump. I get that. And also it's exacerbated, I think, because in academia, there's been a bit of maybe a tightening, particularly maybe towards method developers. I don't know. Like it's almost, and that's what VJ was saying. We had a little discourse on X or Twitter that it's like, he thinks, you can just, Vijay Pandey, sorry.
Yeah.
That you can do everything that he kind of said, everything you think you're doing in academia, you can do at 10x or 100x pace here. Like you should consider it as a jump that you should make. And, I don't know. I think that would be maybe an interesting subject for like a group podcast. You could get a few of the folks who jumped and a few of the folks who stayed behind and really discuss like, what people are thinking about the future and, training of people and that whole interplay. But right now, there is an enormous amount of hype, but also probably there's hype because in principle, this machine learning and AI together with amazing data sets should absolutely transform biology and chemical small molecule discovery, definitely as we know it. But, there's a lot of work we got to do to get there.
Oh, yes. Absolutely. We had a conversation with Paul Robustelli about a similar topic because he was switching back and forth between industry and academia. His point was that, it's good to have both because it's like you can keep the best of two worlds, but you don't need to fully commit to the advantages or disadvantages of either.
Absolutely.
Right? And like the more we can make this barrier kind of available for people to pass through both ways, the better for the field in the end, perhaps. That was his take.
I tend to agree with that. Yeah, I mean, I do tend to agree with that. I think similarly to like this principle that diversity is a strength, it plays to that sort of same theme. And I do think that I guess sometimes I get worried, like if too many people jump, like we don't want to just like a mass exodus of all of the intelligence in some sense, in a particular field out of academia. In particular, also because academic institutions move really slowly. So that it's if you have an exodus, it's areas that have taken kind of years to build up. It takes a while to you lose it, it takes a while to get it back. And so I don't know. Maybe that's just old person thinking.
So many smart people who take their very field-specific knowledge to intellectual property, protected environments.
It is different. Mm-hmm. Anyway.
Okay. Well, I hope someone thinks deeply about these issues. And as you say, maybe it's a good moment, a good thing to do to think about a panel or a broader discussion about it.
Yeah.
Okay.
Yeah.
So anyway, thank you for the thoughts, for the story, for all your insights. Rommie Amaro, it was a pleasure to talk to you.
Yeah, this was really fun. Thanks for having me.
And have a great day ahead of you.
You too.
Thank you for listening. See you in the next episode of Space Invaders.